A vaccine to prevent coronavirus disease is perhaps the best hope for ending the pandemic. Currently, there is no vaccine to prevent COVID-19, but researchers are racing to create one. While vaccine development can take years, researchers aren't starting from scratch to develop a COVID-19 vaccine. Past research on SARS and MERS vaccines has identified potential approaches. Coronaviruses have a spike-like structure on their surface called an S protein. The S protein attaches to the surface of human cells. A vaccine that targets this protein would prevent it from binding to human cells and stop the virus from reproducing. Past research on vaccines for coronaviruses has also identified some challenges to developing a COVID-19 vaccine, including ensuring vaccine safety. Several vaccines for SARS have been tested in animals. Most of the vaccines improved the animal's survival but didn't prevent infection. Some vaccines also caused complications, such as lung damage. A COVID-19 vaccine will need to be thoroughly tested to make sure it's safe for humans, providing long-term protection. An effective COVID-19 vaccine will need to provide people with long-term infection protection. After infection with coronaviruses, reinfection with the same virus is possible after a period of months or years. Protecting older people. An ideal COVID-19 vaccine would work well for this age group. People older than age 50 are at higher risk of severe COVID-19. But older people usually don't respond to vaccines as well as younger people. Health authorities and vaccine developers are currently partnering to support the technology needed to produce vaccines. Some approaches have been used before to create vaccines, but some are still quite new. Live vaccines use a weakened form of the germ that causes a disease. The term weakened means that the vaccine's ability to cause disease has been reduced. Live vaccines are used to protect against measles, mumps, rubella, smallpox, and chickenpox. As a result, the infrastructure is in place to develop these kinds of vaccines. However, live virus vaccines often need extensive safety testing. Some live viruses can be transmitted to a person who isn't immunized. This is a concern for people who have weakened immune systems. Inactivated vaccines use a killed version of the germ that causes a disease. This kind of vaccine causes an immune response but not an infection. Inactivated vaccines are used to prevent the flu, hepatitis and rabies. However, inactivated vaccines may not provide protection that's as strong as that produced by live vaccines. This type of vaccine often requires multiple doses followed by booster doses to provide long-term immunity. Producing these types of vaccines might require the handling of large amounts of the infectious virus. This type of vaccine uses genetically engineered RNA or DNA that has instructions for making copies of the S protein. These copies prompt an immune response to the virus. With this approach, no infectious virus needs to be handled. While genetically engineered vaccines are in the works, none has been licensed for human use. The development of vaccines can take years. This is especially true when the vaccines involve new technologies that haven't been tested for safety or adapted to allow for mass production. Why does it take so long? First, a vaccine is tested in animals to see if it works and if it's safe. Next comes testing in humans. In Phase 1, clinical trials evaluate the safety of the vaccine in humans. This testing must follow strict lab guidelines and generally takes 3 to 6 months. The manufacturing of vaccines also must follow quality and safety practices. During Phase 2, the formulation and doses of the vaccine are established to prove the effectiveness of the vaccine. Finally, during Phase 3, the safety and efficacy of a vaccine need to be demonstrated in a larger group of people. Because of the seriousness of the COVID-19 pandemic, vaccine regulators might fast-track some of these steps. But it's unlikely that a COVID-19 vaccine will become available sooner than 6 months after clinical trials start. Realistically, a vaccine will take 12 to 18 months or longer to develop and test in human clinical trials. and we don't know yet whether an effective vaccine is possible for this virus. If a vaccine is approved, it will take time to produce, distribute, and administer to the global population. Because people have no immunity to COVID-19, it's likely that 2 vaccinations will be needed 3 to 4 weeks apart. People would likely start to achieve immunity to COVID-19 1 to 2 weeks after the second vaccination. A lot of work remains. Still, the number of pharmaceutical companies, governments, and other agencies working on a COVID-19 vaccine is cause for hope.